Tran, the science man. Tran. Brian Tran, the science man. Replication rules. Tran. Brian Tran, the science man. DNA is a double helix.
Brian Tran, the angry science man.
DNA replication, it's important. It's the reason why you're even here. And it's not just you. Every living thing needs to replicate its DNA. It's essential to life. Take a look at this. This here is a microscope of science. Now let's unwind the helix and see exactly what happens during the essential process of replication. Here we have an unwinded DNA double helix, which is consisted of nitrogenous bases that have hydrogen bonds, and there's guanine, which pairs with cytosine, and adenine, which pairs with diamine. So in DNA, there's directionality. So there's 5 prime, which goes to 3 prime, and on the opposite end, there's 5 prime, and there's 3 prime. Now that we've established directionality, let's get into the details. That was helicase. And helicase is an enzyme that breaks the hydrogen bonds and splits the DNA apart. As you can see here and here, these are single-stranded binding proteins which pull the strands apart, which they are trying to get back together. This is primase, and it adds a primer to the DNA strand to start the replication process. This is DNA polymerase, and DNA polymerase adds complementary base pairs to the parent strand, which makes a new strand, which is called the daughter strand.
Go.
This is RNase H, and it removes the primer on the DNA strand. So the process keeps repeating until the new daughter strand is completed in replication, making a semi-conservative DNA strand, and that is the leading strand. On the lagging strand, the process is repeated, but instead it is done in fragments called Okazaki fragments. As you can see here, this is one Okazaki fragment, and that is the other Okazaki fragment. The DNA polymerase just added the new bases, but we still need another enzyme called ligase to glue the bases together. Here you can see this is DNA ligase, and it reanneals the 2 bases together. As you can see here, these are the 2 strands being supercoiled. That was topoisomerase, and it just cut the strand and for it to not supercoil.
Try this at home. It's easier to understand supercoiling when you have visual. Here, we have a phone cord. This is what topoisomerase does to prevent supercoiling. This way, it is easier for the helicase to unstrand the double helix.
Replication. It's DNA, let's replicate. Double, there's 2 new strands. Replication. There's a daughter strand with a parent strand. It is the same, not different. Replication.
Hey Elma, this is me at 12 o'clock in the morning doing this assignment, and me and my dog say stop. There's no freshman beyond this point. Enjoy the bloopers. Okay. We're super cool. Oh shit!
This is RNA. This is RNA. This is RNA. Replication. It's important. It's the reason why you're even here. And it's not just you. Every living thing needs
these nuts. Got him! Got him!
Please give us an A.